Hello, everyone, and welcome to today's OTS webinar series. We're really excited to have Dr. Shalini Andersson, who will present today. She's the Vice President of Oligonucleotide Discovery and Discovery Sciences at AstraZeneca. And also, she started out as an organic and analytical chemist. So she has some really cool perspectives that she'll share with us today. And I'll just hand it right over to you, Shalini, when you're ready.
All right, so good morning, good afternoon, good evening, wherever you are. And I'm really excited to share some of our thinking with you from AstraZeneca. And so I will focus today on trying to get across what are the real challenges and opportunities when it comes to oligonucleotides and developing them into therapies. So in AstraZeneca, we really try to focus on early detection, diagnosis, and intervention when it comes to the biggest challenges in healthcare, and trying really to shift ourselves from symptom management to slowing and stopping disease, so really trying to cure disease. And to be able to do that, we have in the last decade or so, invested very heavily in opportunities to identify novel drug discovery targets. And that we have done through investments in genomics, in functional genomics, knowledge graphs, all to really gain information across the omics area. And I use that to identify novel drugs. So understanding the disease really to identify new targets drugs can then also be defined, or you can do that by utilizing all the big data you generate to the omics platforms, both based on the human data, but also from your models, be it zebrafish or mouse models or higher species, and also the cellular models. But all of this data, by applying AI, you can actually then also identify novel targets that you would not have been able to do otherwise. And to really exploit that you have now access to novel targets that are relevant for human disease, where you can identify models that are cellular or zebrafish or mouse or other species, which are really going to be a way of testing your hypothesis so that you can develop your oligonucleotide drugs into therapies. We have, as a company, also invested in a lot of different modalities, including oligonucleotides, to be able to modulate the novel targets with the right modality, so that you are actually using the right kind of molecule to modulate your target in the best possible way. And so, as you can see here, we have, as a company, invested in small molecules, which is our core business, and antibody therapeutics, which is also our core business. But in the recent years, then we have expanded into cell-based therapeutics, peptide and proteins, and nucleotides as well then. So if we focus on oligonucleotides, then you can divide the oligonucleotides into major baskets, you could say. So you have a group of oligonucleotides that have a design such that they require hybridization, so to be able to act upon the RNA, or you could say that they are hybridization independent, and then they can actually work on their own without having to hybridize with the mRNA. And you have a number of different flavors here, so like everything from siRNA, microRNAs, antisense oligos, CRISPR-Cas for gene editing, etc. And then you have the hybridization independent ones, where you have aptamers, mRNAs, immunostimulatory oligos, self-replicating RNAs, DNA, etc. So there is a lot of opportunities when it comes to oligonucleotides as therapies. Now, if we focus on oligonucleotides where you want to up or down regulate protein expression, then you could say that you're using, normally what you're doing is that you have your DNA, which is transcribed into an mRNA, which is translated into a protein. And so if your protein is disease-causing, then you want to try to block the translation from mRNA to the disease-causing protein. And then you can, for example, then use antisense oligos or siRNAs to do exactly that in a very efficient manner. However, oligonucleotides, as I already said before, are extremely useful and versatile molecules, and you can have a range of different mode of actions going for a target either in the cytosol or in the nucleus, which really gives us a lot of opportunities to modulate targets that can be modulated by oligonucleotides as therapeutics. So there is a range of possibilities which actually not really exploited yet, I would say. So there's lots of opportunities remaining, and it all depends on the target, the therapeutic target, or the disease-causing target, or the disease target that you're really going for. However, as you can see here, during the last years, you have had a number of approved drugs when it comes to nucleic acid therapeutics, and it's been an increase in the number in the last years with a lot of different approaches, so different mode of actions, different chemistries, different tissues that you are targeting, and also different kind of oligonucleotides. And so this area is really expanding very rapidly, and you have more than 100 nucleic acid drugs currently in clinical development. So there's a lot more to come, and I think what we see now is that the belief in that these molecules have been clinically validated as a modality, and therefore we feel more comfortable with the chemistries we are using, we feel more safe, and we can also design them better than we used to be able to do. However, these molecules come with their challenges, like all molecules, I would say, when you want to develop them into therapies. And for oligonucleotides, if we look at them, they are extremely poor when it comes to drug-like properties if you were not to modify them. So if they were natural nucleic acids, because they are highly, highly charged. They are rapidly degraded by nucleases in plasma and tissue if you were to apply them. They're very poor pharmacokinetics, and they're cleared rapidly by the kidney. So you would need to dose several times a day to really, or even hourly, to really get them to work. They have really not so high affinity for your target RNA. And then they also can cause immune stimulation. So it's not a very good molecule per se. However, it has good possibilities to be modified. And there's been a huge amount of effort in the last 30 years that has gone into trying to optimize, modify chemically the backbone of the oligonucleotides, the 2' sugar modifications, as you can see here. There's been a lot of work done in trying to enhance the nuclease resistance by designing the oligos right with the chemistries that are more resistant to nuclease attacks. And there have also been a lot of effort going into enhancing the affinity for the target mRNA, which is your target that you want to actually degrade or modify by splice switching or things, or block certain areas in, etc., depending on the mode of action you're trying to go for. There's also been a lot of understanding on the binding to proteins in circulation and how you can design and your chemistries and your oligo design to improve the pharmacokinetic properties and enhance safety and tolerability. So a lot of effort is going into to get us to where we are today, where we actually have a drug molecule with chemistries that are pretty well understood, where we understand what drives the efficacy, and in quite a lot of aspects, also the safety of these molecules. So if we have come past by the big challenges of the chemistries and having the drug-like properties of oligonucleotides, there still remains the other big challenge, and that is they remain highly charged. And so really they are big molecules, really highly charged, pretty hydrophilic, and not very much designed to enter cells. There is a barrier that they have to overcome. And you can do that in several different ways. You can do that by passive biophysical targeting, so using lipid nanoparticles that can cover and encompass your oligonucleotide and thereby enter cells. Or you can have dendritic polymers and such, which are more what I call passive biophysical targeted delivery of your molecule to the cell. Or you can go actively and target your molecule through conjugation to a ligand that's going to help you enter the cells. So there are different approaches to this. And from my perspective, I think for the shorter oligonucleotides, such as antisense, oligonucleotides, siRNAs, and so forth, I think what we have seen is a shift towards trying to move towards targeted delivery of these oligonucleotides using ligand-mediated targeted delivery. So by using ligands of different kinds, so sugars, fatty acid, aptamers, small molecules, antibodies, peptides. So you can use a variety of these ligands that bind to cell surface receptors and can help internalize the conjugates, which is this part, into the cells. They will then you will be able to deliver them to specific cells in specific tissues. And if you can do that very, very efficiently, then you will be able to modulate new targets in those tissues and cells, which have not been able to modulate before. And that also, by targeted delivery to specific cells and tissues, you reduce the dose and the cost of goods of these molecules as therapeutics, because you will not need to have a systemic exposure, but can actually really direct your drug molecule to the cell type and also the tissue in which you want to modulate your target. Having these kind of ligand conjugated to your drug oligonucleotide also allows you to get proprietary IP. So patent filing is possible for you to own a patent around your molecule. So that is not a small thing or a trivial thing when it comes to developing drugs that you want to be able to get to patients and also to the market. However, you might not always need to have targeted delivery in the sense that you need to conjugate to a ligand or have a lipid nanoparticle or a polymer that helps you deliver to where you want it to go. You can have sort of a targeted delivery to certain compartments, which are quite amenable to that, and that there is direct administration of your oligonucleotide to compartments such as the eye. So that's a pretty much contained compartment, or the CNS or the skin. And so you can have local distribution of your oligo for those kind of compartments. So that's a possibility to directly inject into those compartments. Now, what are the real challenges also with, it sounds a bit simple. Yeah, you have a ligand, you conjugate it, and you can deliver it to wherever you want. Unfortunately, it is not. This is really the big challenge. So when you have a drug molecule, a small molecule, for example, you actually are focusing very much on trying to optimize the drug molecule. And that's a huge undertaking and super challenging. In this case, we are really trying to optimize 3 molecules in one. So you have your drug molecule, which is your oligonucleotide. So you optimize that to be able to modulate your target. But linker, and you also need to optimize then the targeting ligand. And you have to balance all 3 of them in the same molecule together. And that is really challenging. And it sounds not so difficult, but I can promise you it is really challenging because there are a lot of different aspects you have to optimize. So in principle, you could say that if you, for example, then use the GalNAc ligand conjugated to an antisense oligo, which I only show in this paper, you can actually reduce you can enhance your potency tenfold and thereby reduce the dose you have to administer to the mouse, but also to patients, which is what you can see here in this slide, where I only showed very, very nicely that if you have your naked ASO and you conjugate it to a GalNAc, you will be able to reduce the dose tenfold. And this works very well in healthy volunteers, as you can see here. And this they have done for a number of different antisense oligos. So it's something that works more broadly, you could say, for oligonucleotides, if you want to target a biological target that you know is disease-causing in hepatocytes in the liver. So it's an extremely efficient way of doing that. Another example is really the biodistribution or changing the biodistribution of oligonucleotides by conjugating to lipids. So by using different hydrophobicities of these lipids, so how hydrophobic or hydrophilic they are, you can actually distribute them differently. And there are a number of different fatty acids that have been, or lipids, that have been used for this purpose, such as fatty acids, vitamins, and sterols. And then quite a few papers around this, as you can see below here, Biskans and Osborne as well. And what you can see here is that, and this is what the paper from Biskans has shown, is that if you increase hydrophobicity, you are going to change your distribution to the liver mainly. And if you have less hydrophobic lipids, you're going to distribute your oligo to mainly the kidneys. And so by modulating the hydrophobicity of your lipid, you can actually modulate where it ends up more or less of. So that's a way of trying to modulate the distribution of your oligo through the hydrophobicity of your lipid conjugate, so to speak. One other approach where we have been more active on and where I've been very much interested in is really targeted delivery of antisense oligos or siRNAs to pancreatic beta cells. And this was very much driven from our interest in trying to find ways to modulate targets that are of interest to us in the pancreatic beta cells. Now, whilst antisense oligos have a very broad uptake and distribution in in vivo, in animals and in humans, they are really not taken up by pancreatic beta cells. They are pretty refracted from that. So from our point of view, we really wanted to be able to find ways of getting an oligonucleotide to the pancreatic beta cell. And the reason for that is really when you have type 2 diabetes and you increase your weight, you could say, okay, I dropped that as well, my pointer, so let's get that. So when you become more obese, you are going to really crave that the islets expand and produce more insulin to manage the sugar levels in your body. So what happens then is that the pancreatic islets at the end sort of close down because they get fatigued because of the amount of insulin they have to produce. And then you sort of will have diabetes because you can't produce enough insulin. So what we have done is to really identify GLP-1 peptide, which can conjugate it to MALAT1 antisense oligo, could then be shown to be able to knock down MALAT1 gene in beta cells. And what you can see here is that this is when you have administered only saline. This is the naked MALAT1 antisense oligonucleotide, and this is the conjugated oligonucleotide. And as you can see here, the pink dots are actually the mRNA of MALAT1, and as you can see here, you have knocked down most of it. So this actually tells you that you can deliver the oligonucleotide inside the beta cells, and MALAT1 antisense oligo can bind to the mRNA and degrade it, which is really cool and something that is then can allow you to design oligonucleotides towards drug targets and do something similar. And what you can see here is that this just not works in cells, but also in primary mouse islets, where you can see that we by treating these mouse islets with GLP-1 MALAT1 ASO, we could knock down MALAT1 in those primary mouse islets. And also we use another antisense oligonucleotide for FOXO1, which is also translating to protein knockdown. So not just because MALAT1 is a non-coding RNA, so you can only see the knockdown of the mRNA. But if you want to see that you are not just knocking down the RNA, but also then you are getting reduction in protein, which is what you want to inhibit the production of, because it is your disease-causing protein, right? So that's what you can see here, that also for FOXO1, we are able to show that we can knock down FOXO1 in mouse islets. And then we move this into in vivo setting. So you could see here that we are getting MALAT1 knockdown in the islets in vivo, in mouse. And the same you can see here in immunohistochemistries, that you are knocking down GLP-1 MALAT1 ASO in the islets, which is really a very good proof of concept that we did at that time. Now, when I said that it is really significant that you be able to optimize both your drug molecule, which is your oligonucleotide, but also the linker chemistry and the GLP-1 peptide in this case. So you are trying to optimize 3 different molecules in one. Oops. And that of course is very, very challenging. And so what you can see here is that depending on the linker you use, you will get different gene knockdown. So it is really, really critical that one optimizes the linker chemistries as well. It's just not about optimizing your drug molecule and the ligand that you're using to conjugate, but also the linker in between. And it has a huge impact on on ED50, so the efficacy of your molecule. Now, if we look at AstraZeneca per se, we are focusing mainly on some of the areas when it comes to therapies. So it's oncology, cardiovascular, renal metabolism, respiratory and immunology, and vaccine and immune therapies. But also recently, we have entered into the area of rare diseases. And we have access to all kinds of modalities and platforms to really enable us to modulate targets in all these disease areas that we are interested in. And so I'll walk you through a couple of examples where we're using oligonucleotides to modulate these targets. So I'll start up with one of the targets that we have in phase 2 now, which is for NASH, so non-alcoholic steatohepatitis, which is a leading cause of chronic liver disease and is extremely difficult to treat today because there's really no approved drugs for this kind of disease. So NASH is really something that occurs. It's a progressive disease, which is not so easy to diagnose early on. And it moves the liver from being steatotic to non-alcoholic steatohepatitis, and the end stage, which is cirrhosis. And that is really going into liver failure and transplantation kind of setting. So it leads often to then death. And so if you think about it, there is a target that we identified as PNPLA3 148 mutation, which leads really to impaired lipid handling and non-alcoholic steatohepatitis. And so what happens is when you do have this mutation, you actually are not able to handle lipids very well, and you will get these enlarged lipid droplets in your liver. And what you call NASH liver with a lot of inflammation and fibrosis. And so by treating with the PNPLA3 directed ASO, we have been able to show that we can reduce NASH and liver fibrosis after treatment, and you get more normalized lipid droplet sizes in the liver and a more healthy, less inflamed, less fibrotic liver. And so with that hypothesis in place, this is a molecule that we have shown works in mice, in humanized knock-in mice with PNPLA3 gene knock-in. And we have been able to show that with the control ASO, you have lots of inflammation, lots of fibrosis and steatosis with lipid staining. And when you have treated these mice with PNPLA3 ASO, you reduce a lot of that. And you can also see here in the scoring that we have the NAFLD score, which shows you that you actually improve the health of the liver in these mice as well, even though they are then even though we have induced NASH in these mice to start with by the feeding they get. So that was our starting point, and this now molecule, of course, has gone into man. And as I said before, it's in phase 2. So it's a very good example of really doing precision medicine towards a specific gene using a precise modality with the GalNAc conjugate, which is really delivering the ASO to the hepatocytes in the liver. And of course, then that hopefully, we hope that this will actually offer this kind of patients disease resolution going forward. And it looks promising at the moment, but well, lots to see yet. The other example I have is around dyslipidemia, which is also a very serious medical condition. These patients have highly elevated levels of LDLC, which is a high risk for cardiovascular disease. And there has been a clear causative link shown between LDLC and MACE. Normally in these patients, you can measure the LDL-C levels, and that is sort of recognized as a surrogate endpoint for disease. PCSK9, I think many of you have heard about this story. You have monoclonal antibodies that bind to the extracellular domain to PCSK9 and therefore can block that and reduce the ability to shuffle the LDL particles. What you also have is a short interfering RNA, Leqvio, which is on the market and also known as inclisiran, which is inhibiting PCSK9 translation into the liver by acting on the mature mRNA. So in principle, binding to the mRNA for PCSK9 and thereby stopping the translation of to the protein. And of course, that means that you're going to, in the end, reduce the LDL-C levels, which is what you want for these patients. And this actually works very well because the target is in the liver hepatocytes. And what you're doing is using the GalNAc conjugated oligonucleotide. And in this case, when it comes to antisense oligonucleotide, and in inclisiran's case, it is actually an siRNA, so it's a double-stranded RNA. You are using this to deliver the antisense oligonucleotide to the hepatocytes in a very precise manner, and that is actually acting on the mRNA. And in this, since it's an antisense oligonucleotide, is also acting on the pre-mRNA. And so you're reducing the levels of PCSK9 protein that would have been produced because you are actually degrading the mRNA levels. All right, so as I said already, the AZD8233 is an antisense oligonucleotide that's conjugated to GalNAc to deliver an oligonucleotide that will reduce PCSK9 protein levels in hepatocytes and circulation. So this is a good example of how you can conjugate GalNAc to more broader diseases, not like the previous example of NASH with the specific mutation of a gene, but rather quite a huge population that is not being able to manage the LDL-C levels. And therefore, we are talking here about millions of patients. It's not a rare disease population. But you can see that if you have targeted delivery that is efficient, you can actually modulate a broad number of diseases. And that is what you also see in the clinical trials. Now, there's a huge amount of targets that are liver targets that are involved in disease that have not been able to be treated before, where by using GalNAc as a ligand, you can now start to deliver different kind of oligonucleotides to the liver hepatocytes to modulate those targets and thereby develop drugs that can really help patients who have been untreated up to now or treated not efficiently up to now. So this is some of the data we have from the clinical trials. So you can see here the subcutaneous administration of AZD8233 really reduces the plasma levels of PCSK9 proteins in humans. And these are the patients that would be normally helped by getting this kind of reduction from when it comes to the cardiovascular risk that they are running with. And it's really very efficient. And you can say that this is like every 40 days or every 30 days, you would be giving them a monthly injection, and then you would be able to keep this at steady state at a lower level. And what we see from this is exactly that you will be able to reduce the circulating PCSK9 levels to less than 90% of what they were. And this was really going to be efficacious in case this now does show that this is the case also in the later stages of the clinical trials. What we also see that the reduction of LDL-C, which is, as you remember, the biomarker that you measure in these patients to show that this is having impact on the disease. You want to see the durable reduction in LDL-C, that you have that as well, because this, you can see LDL cholesterol on the y-axis, and you can see that you're getting a really good reduction in LDL-C levels with the 90 milligram dose. And so with that, I would just like to acknowledge the disease area teams that I have been working on these projects with, and also Ionis for developing these molecules together with AstraZeneca in collaboration. And with that, I apologize for all the technical problems, but I hope that I can take some questions as well.
Yeah, thank you, Shalini. Thank you for this excellent presentation. I really enjoyed it. So if you have a question for Shalini, please use the Q&A function. And I will kick off with a few questions. So I think you mentioned, and I think we agree on that, delivery is one of the most challenging things in the oligo world. So I was just wondering, what do you think is the most challenging organ to deliver these kind of compounds? And my follow-up question, or the second question is, I think you mentioned the GalNAc, and this is very efficient in delivering compounds to particular liver cells. But do we need other compounds to target other cells that are present in the liver?
Yes, I think the one area which is super challenging, I would say, is the CNS. I mean, unless you want to do intrathecal injections directly, which is really not maybe what you would want to do for all your life. I think that's very, very challenging. And it would be fantastic if we could find ways to deliver to the CNS and the brain. I think what we see, as I said before, when you do find a very efficient way of delivering to specific cells, like GalNAc is for hepatocytes, you really open up the target space. So diseases like NASH that have absolutely no medications today that help, those are now, those will, I hope, soon get treated, and the patients will hopefully feel much better and also survive longer than they do today. So I think there are a lot of these tissues and cells that we and others are working on, because we know there are a lot of very novel and very good targets, but we just can't modulate them in an efficient manner. And so I would say, for me, the kidney, the lung, the heart, and the CNS, and actually muscle, although we have quite a few muscle dystrophy oligonucleotide drugs on the market, it's still not very efficient. So if you could find ways of really delivering these molecules efficiently to these major organs, we would be able to develop a lot of new medications that could help patients who have absolutely no other options today.
Yeah, and then my follow-up question would be, so I think one second thing that's also super important is the safety profile. So when you work with all these conjugates, with all these linkers and the new combination of conjugates and linkers, do you have any idea about safety profiles, or do safety profiles change, or anything known about that?
Yes, I mean, you would have to treat them as, so if you think about the targeting ligand, you will have to take your targeting ligand on its own into a safety assessment, so both in vitro and in vivo. And then you'll have to do the same with your oligonucleotide, which you would do during the development of it as a drug molecule. But then you'll have to take the whole molecule, including the linker, into a safety assessment, because you are going to have that as your drug molecule as a whole. So you will have to do safety assessments for all three, you could say.
Okay, so we have a whole bunch of questions in the chat. We have 19 so far. So maybe if you can answer them short or relatively shortly, then we can handle quite a few of these questions. So first question is, I think a former colleague, Jimmy Waterings. He asked, in terms of endosomal escape, should you aim to increase release by yes or no, damaging the endosome?
Yeah, well, there are many people who have tried also that, and we are also looking at it, and we published a little bit on that, on using endosomal escape enhancers. In our case, these are small molecules that we have identified through our by doing an HTS, so high throughput screening, sorry, of our drug collection. To see whether we can identify molecules that could aid in that, and that seems to be promising. And of course, that's what Jimmy says is that this is one way of increasing the dose, take the dose inside the cell, so that you can actually have your molecule be able to do what it should do as a drug, rather than be trapped in the endosome and then go for degradation. So yes, there is some effort going on that, and I think others are also doing that.
Okay, question from Shalini. Is there a way to rationally design linkers for oligonucleotide conjugates? Not in our hands, at least. We are looking at a broad number of linkers, and this is also where a lot of effort is going in, because that's how people, if you look at the GalNAc conjugates, there are more than 10 companies who have their own GalNAc conjugates. The GalNAc ligand that binds to the acylglycoprotein receptor is the same. So it's the galactose sugar molecule, but the patent space that different companies have is all based on linkers, and that doesn't seem to be so critical, because if you look at all the so we have looked at 6 or 7 different GalNAc molecules, that the only differing part is the linker from different companies, and you really get similar knockdown. So it seems to be quite accepting of structure changes there. So it's not so critical. The critical part is the ligand, the GalNAc ligand, and your oligo. So you can do some changes there without really impacting the potency of your molecule, but you still need to optimize it. So together, how they, and I think I showed that with the GLP-1 conjugate, that it does matter what your linker looks like. So you need to optimize that together with your oligo and your ligand. Yeah, and then a follow-up
question. To have a rational design, I don't think so, not yet.
Then a follow-up question from Pedro. Where are you seeing the highest impact of the linker? Is it on plasma stability, circulation time, intercellular antisense mechanism per se? So any ideas on that?
What we have seen is you can impact that on potency mostly. Circulation time you can impact by doing peak extensions if you need to, like having a lipid molecule conjugated to that somewhere as well. You can do that just like you do with peptides when you prolong the PK and circulation time. I can't say there's a rational design for understanding the linker and endosomal escape at the moment. It's just not in our hands.
Okay. Let's see. So next question. Also another question from Pedro. So I think I've seen a few modalities where you can have like where you can link 2 or 3 or multiple ASOs. So do you see room for 2 or 3 or multiple ASOs targeting different genes simultaneously in the same cell in diseases of high complexity? So for example, cancer or neurodegenerative disorders.
So I see that, I've seen that as well, and there's always a discussion around this, because when you have several different oligonucleotides for different diseases, what you then locking yourself into is that you will need the same dose to modulate all the 3. Say you have 3 oligos for 3 different targets, then you are locking yourself into saying you're going to need the same amount or the same dose for all 3 diseases in a variation of population. I think that's really, really challenging, that you lock yourself into that, because you will have locked yourself into 3 mg or 10 mg or 15 mg or 25 mg or 100 mg a kilogram, because you don't really know that that is what you need to modulate all 3 targets. So I'm a bit skeptical about that, but that's my view.
So then we go to the GalNAc conjugate. So I will combine 2 questions. Is there a better option than GalNAc? And then I will combine it with, could you please comment on the advantages of GalNAc conjugated to ASOs over GalNAc conjugated to siRNAs?
Ooh. So better than GalNAc has not really been identified for hepatocytes yet. There have been a lot of different activities to identify that, but I would say it's one of the more efficient ones. So if there are other cells in the liver that need targeting, yes, we need more work to be done there, because you have a lot of targets in the stellate cells, for example, for fibrosis and so on, that are not being able to address today because we don't have an efficient way of delivering to stellate cells in the liver. The other question was around GalNAc conjugated to ASOS or siRNAs. What are the pros and cons there? Well, I mean, the pros for siRNA is that GalNAc conjugates of siRNA do have longer duration, because siRNA as a drug molecule has longer duration. So you are going to need less frequent dosing. So you could have patients taking every 3 months or every 6 months. There's also been some papers around being able to extend it to beyond 9 months even. So of course that means that the patients do not have to take this very often, compared to antisense oligos, which have been shown to be in the range of between 2 to 6 weeks, or maybe 8 weeks at the as the most period I have seen. But on the other hand, one should not forget what that means. It means that the patient has to remember every 6 months I have to take this. It's not always compliance of the patients is not very easy when they don't do things on a regular basis. So the jury is out there. I would say there are too few examples on the market yet, and we have yet to learn from how the patients use these drugs before we can say what is the best thing to do. But obviously the duration has an advantage for siRNAs in many cases.
Yeah, so you mentioned that delivery is one of the biggest hurdles in oligonucleotide development. So what are the other hurdles? Or are there any other big hurdles?
Yeah, there is, because if you think about antisense oligonucleotides, which actually one should not forget, those are molecules that are really your option to go with if you're going to use them for splice switching or blocking mRNAs and things like that. there's a very good uptake of these molecules, but we don't understand the safety challenges of these molecules when you go to higher doses. So we know that if you go to doses of about 250, 300, you are bound to get some thrombocytopenia and things like that. So you try to keep out of that range. And therefore, targeted delivery becomes very important. So I think if we could improve the safety profile of antisense oligonucleotides by designing them differently or different chemistries that could be more efficacious, and therefore you would not need higher doses, could be a good thing for the future.
Yeah, and maybe to relate on that, because I think what you can assume is that you need an application that would need chronic administration. So do you have any thoughts, or maybe is AstraZeneca working on it, or what is your opinion on the activity investigating stereo-pure antisense oligonucleotides?
I think the jury is out there on the value of that. So I think more is around, from a patient perspective, the sub-Q administration is a pain. So we are putting some effort in trying to see if we can develop oral oligonucleotides so that you could actually take that as some sort of a pill. It's still going to be very challenging, but I think that's more where we are going rather than stereo-pure isomers at the moment, from our perspective.
Yeah, and then it's around the hour, so I will do 2 more questions, if that's okay with you.
Yeah, sure.
So, because there are still many more, but we cannot handle all of these. But I will go on with a question from John. Also, it's about the future of oligonucleotides. So, do you think they will be more dominant than therapeutic antibodies? Because since especially in the beginning of the presentation, you mentioned that AstraZeneca also is a big player in that field.
I think that we will see a larger proportion of drugs being oligonucleotides going forward, because they are very useful molecules and they are versatile in that you can modulate targets intracellularly. So there are different mode of actions. Antibodies will be per se going for the targets which are on the surface of the cells, whilst getting intracellularly is going to be more difficult for them. So I think you use the right modality for the target that you want to modulate, such that you get the most efficient modulation of the target. And then it doesn't really matter what it is. But absolutely, I believe that we will have much more nucleotides as therapeutics going forward. The more we learn, the more we understand how to use them best. And if we can get more of the targeting approaches in place, then it will expand the druggable target space and with nucleotides.
Yeah, and then the last, I think, is also a very general question that might be quite challenging to answer, but just trying to go for it. So, what are the key criteria for choosing the targets for oligotherapeutics?
Ooh, that's not very easy to answer.
We end with a difficult one.
Well, you could say one of the key criteria is if it's an intracellular target and it's best, and if it's a protein which doesn't have a pocket where a small molecule would be able to be designed for, then it's very, very challenging to design a small molecule for that. And I would say, yeah, then oligonucleotides would be a much better option.
Good. Well, once again, thank you very much, Shalini. I really enjoyed the webinar. And also, I think we had a pretty good Q&A session. So if you still have a question for Shalini, you can email us at info@oligotherapeutics.com. So then you can direct it to Shalini.